Hello, welcome to the lecture about gene therapy. I'm Dr. Stephen Brown. I'm a lecturer and RNAi Screening Facility Manager at the University of Sheffield in the School of Biosciences. Today's lecture, I'm just going to run through some little bit of information about research that we do and some of the things that go on in Sheffield. Then I'm going to cover gene therapy and the types of gene therapy that are available and the way that we can get it into patients. And then we'll have a little bit of a recap at the end. Okay, so the University of Sheffield is first at the moment in the UK for medical research work as assigned by the government in one of their research exercises. We've got a very good students' union, very friendly place, it's very green. The university campus is in the centre of Sheffield, and we're top 5 in the UK for biological sciences. So all round a quite a nice, interesting place to learn and to study at. You may be asking, where is Sheffield? So Sheffield is in the centre of the UK. We're surrounded by Manchester, Leeds, and we've got the Peak District very close to us. So there's lots of walking and hill climbing sort of activities available, and we're only 2 hours on the train to London. So at the University of Sheffield, we do lots of interesting research work in terms of biosciences. My research work is mainly interested in RNAi screening and the use of RNAi as a therapeutic. And we'll cover a little bit later about the use of RNAi and other technologies for gene therapy. But as well as my research work, there's some interesting research work that's quite groundbreaking at the University of Sheffield. Involved in evolution, sustainability, biodiversity, so sort of ecology and conservation type subjects. And then we also have quite a nice biotechnology aspect of our biosciences, where we look at food security, CRISPR, antimicrobial resistance for antibiotics, all sorts of things like that, and a big aging and disease grouping as well as research on cancer and COVID and stem cells and all these things that you'd expect from a very large biosciences school. So there's a little bit about the background about me and the university. And let's go now into gene therapy. Now, there's some nice definitions out there of gene therapy, and it might surprise you a little bit about the way that various different governments and agencies think about gene therapy. So generally, when we think about it, we're trying to replace a mutated gene, a gene that has base pair changes that end up and lead to disease. And what we'd like to do is to replace that with a healthy copy of the gene. So that's what most people think about gene therapy. We're replacing a broken gene. But we can also inactivate or knock out a mutated gene, or maybe a gene that's not functioning properly. And there's various technologies that allow us to do that. And in addition to the use of genes to replace mutated ones, we can also use people's cells as well. So we can take cells out of an individual, engineer them, replace them with a working copy of a gene, and then introduce those cells back to the person. And that also comes under the remit of gene therapy. So why is gene therapy important? Why is it garnering some interest at the moment? Well, if you think about it, up until this date, medicine has mainly used surgery, biomedical devices. We've also used natural products. We've transplanted tissues or cells, and also we use chemicals to help us out that solve different diseases. However, quite often those don't lead to a cure for a particular disease. And gene therapy allows us to actually target a specific gene and hopefully will cure that disease. So actually it's more targeted, more specific, rather than this managing of a symptom that quite a lot of the other therapies use or perform. So when we talk about gene therapy, what are we actually thinking about in terms of the material that we use? So generally we've we tend to use these technologies that are listed on this page here. We use plasmid DNA. Plasmid DNA is small circular DNA isolated from bacteria. We also use viral vectors. And vectors or vehicles are the way that we can carry that genetic material into the human cells. So, for example, COVID's quite a nice one recently, where AstraZeneca have used a particular virus to deposit part of another virus inside of us to raise an immune response. And that's quite a tried and tested piece of technology. People also use bacterial vectors. So we can use bacteria to actually deposit genes inside of us. You know, there's enough bacteria in our guts, there's about the weight of our brain. So they're quite good ways of delivering information. And more recently, which there have been some high-profile Nobel Prizes, where you can actually deliver genes through CRISPR technology. So this way that we can actually cut and paste genes into the human genome, or even cut and delete genes out of the human genome using this CRISPR technology. And of course, don't forget, we can also use cell-based therapies as well, where we can take people's cells out, engineer them either with viruses or with plasmids or with CRISPR, and then reintroduce the cells back into individuals. And maybe some of the things that might surprise you is that we also use modifying technologies like RNAi or antisense messenger RNA, or even introduce messenger RNA. And of course, most people now are becoming quite familiar with using messenger RNA as gene therapy because of some of the COVID vaccinations are now using it. And we'll talk about some of these therapies later on. So what stops us just going ahead and doing gene therapy on anyone? Well, actually, there's a gene therapy advisory committee, and that's based within the NHS, and that was set up in 1993 in the UK to regulate the use of gene therapy. And of course, they normally see this therapy that's then presented to them, and then they discuss whether it's viable or not, and then that therapy then gets tested out in clinical trials in hospitals to see if it's acceptable to be used on patients. So let's first consider the central dogma of molecular biology. This is something quite important. So this is the only heavy science part, hopefully, that you'll encounter in this lecture. So I've got a nice little sort of flow diagram here set out of when we think about the human genome and what do we actually mean and what does that relate to in terms of genes. So quite often you'll see this human carrier type on the top left, and that's the way that we sometimes represent human chromosomes. The human genome has 23 chromosomes in it with an XY chromosome, and you can see here that chromosomes are labeled from 1 through to 22 of the autosomals, and you can see that chromosome 1 is the largest chromosome there. So there's about 3 billion base pairs in the human genome, and that was sequenced around about the year 2000, and we've known this information for some time. So in every single one of our cells, we should have the normal complement of chromosomes. And every time a cell divides, it replicates its DNA, and it needs to actually replicate the whole of the genome in a very short amount of time, reproduce it, and then 2 daughter cells arise from that replicating series of genomes. But once the cells have replicated, they need to become functional. So they normally transcribe genes. So the genes are normally expressed, and that transcription normally involves the making of messenger RNA, and those messages then are then translated and then form the protein. So if you fell asleep during this slide, if you looked at the bottom part where it said replication, transcription, translation, and protein, then that pretty much covers the thing that you should take home from this. But if you unravel one of our chromosomes on the top here and stretch them out, and the chromatin are the proteins that are associated with the DNA, and you can unravel those, you should begin to start seeing the structures of genes. You can tell them by the sequences, and each gene normally has exons and can have some introns, and the introns are the components that are never used in the messenger RNA. So when we produce messenger RNA, all the exons are joined together. and that's formed a process called splicing. So the messenger RNA then leaves the nucleus, and then that enters the ribosome in the cytosol or the cytoplasm, and that forms the polypeptides or proteins. So every gene could theoretically give rise to a messenger RNA that gives rise to a protein, and the proteins are the ones that have the enzymes and the capability of doing things. So when we talk about gene therapy, really what we're trying to do is make proteins that do things. So to give you some idea and thought about this, I said there were 3 billion base pairs or in each cell. So if you unwound them and produce a long string of DNA, every cell would be about 2 meters long. And there are about 10 trillion cells in each human. And so if you took my DNA from every single cell and you attached it to the sun and stretched it out across our solar system, you could get to Pluto and back to the sun at least with some to spare from my genome, which is quite interesting. So actually DNA is incredibly well packaged inside each cell. So now we're going to talk about some of the different types of technology we use to get the gene therapy inside. So here we have a rudimentary structure of a plasmid, and plasmids are actually found in bacteria. So lots of bacteria hold them inside, and these are extra chromosomal components of DNA, really. So they have their own bacterial genome, but they also hold these tiny little circles of DNA that they use to help them grow and survive in the environment. And we've actually hijacked them and start put human genes inside of them. We can use those to transfer DNA to patients. But we also use viruses. And obviously most people are quite aware now of HIV, measles, or SARS-CoV-2, which forms COVID. So these are quite familiar and quite widespread amongst us at the moment. But we've also got a number of viruses that are actually quite beneficial for our use. So we've got adeno-associated viruses down here on the bottom, and the one on the left is what they typically look like, but we've now begun to engineer them so we can actually put human genes and host them inside them. So basically using these viruses as a vector to give that genetic information to patients. So just to give you an idea about scale again, so if you think about a grain of salt that you might put on your fish and chips on a Friday night, well, those are about 1 millimeter across. And if you think about one of your hairs on your head, they're about 100 microns, and some people have slightly thinner hairs than other people, but there's a range of about 100 microns for a hair. But then if you consider your red blood cells or other cell types in the human, they range from about 10 microns up to about 40 or 60 on average. Bacteria, again, are about a micron, so we're getting smaller and smaller. And then we think about viruses. Well, viruses are now in the nanometer scale, 100 nanometers. There's quite a lot of viruses. And then we think about proteins and larger molecules that are inside a cell, and they're about 10 nanometers. So you can see that we're talking in a nanometer range now for all the things we'd like to deliver for gene therapy. Now, we've talked about plasmids and we've talked about viruses, how we can use those to give the information to patients, but we can also use mRNA therapies. So basically, mRNA therapies are synthetically produced messenger RNA in the lab, and then we basically form a little shell around them of lipid bilayers. And those lipid bilayers can quite easily be taken up by cells. So we have our little complex lipid nanoparticle here on the left, and they can get taken up by cells, and then they are eventually released and degrade, and they release their messenger RNA into the cytosol, and those are then translated to create a protein. So that's another way of delivering our gene therapy. Now, at the start, I said that I worked on RNAi silencing. So RNAi is actually a natural mechanism where our cells use it every day. Every single cell in my body is using the RNAi machinery because we make our own RNAi molecules, but we've learned some tricks of the trade from our own cells, and we can add some artificial RNAi through these siRNA molecules, short interfering RNAs, and we can use lipid nanoparticles again to deliver those to cells. And once we deliver those to target cells, well, actually they get taken up and they enter something called the RISC complex, and they can actually then target the messenger RNA that has a complementary sequence to our siRNA. So it can base pair with it, and then it can actually cause degradation of our target mRNA. So siRNAs are really good at specifically targeting the mRNA, maybe from a mutated gene, and then destroying it. So we can inactivate that mutant gene. And of course, the one that we're all familiar now with is the CRISPR gene editing technology. And originally, people were using it to delete genes. So CRISPR allows us to almost molecularly cut, like a pair of scissors, a specific region of the genome, and then we can either ligate the two parts of the chromosome back together again, getting rid of maybe the mutated gene in the middle, or we can use it to create a cut and then paste in a gene of interest. And nowadays, people are actually using it to correct mutations in the genome. So there's a whole host of technologies that are available. And of course, you can engineer stem cells or other different types of cells that are useful for gene therapy technologies. So in the real world, what's going on? So this is just a recent example of baby Arthur that was presented in the BBC website and on BBC News. Basically, poor little baby Arthur had spinal muscular atrophy, and that would probably mean in death somewhere in his 20s. And the reality for baby Arthur would have been that most of his motor neuron cells would have actually died without a copy of the SMN1 gene. But basically, they use viral vectors to replace the SMN1 gene into cells that allow the motor neurons to survive and for baby Arthur to start living a normal life. Now that's great news and really lovely example here produced by the Novartis company. But unfortunately, those sorts of therapies are £1.8 million per child, and there are only 40 kids unfortunately born every year. So that creates a bit of a problem really for the companies that are developing this sort of technology. Is it worth their while to make this type of therapy? So what are our limiting factors of gene therapy? So we talked a little bit about the cost of delivering the SMN1 gene. And that's not actually probably the most famous example of gene therapy. In 2017, Kymriah was developed, which is a CAR T therapy, and that basically programs people's T cells, which are sort of white blood cells, to actually target and kill leukemias. And this was developed in 2017, and there are a number of different CAR T therapies now available for different diseases, but this particular therapy costs £280,000 per patient. Now, long term, that might be a saving, because if a patient's on a number of different drugs and long term in hospital, in and out of hospital, that could actually be a saving. So this is something we need to consider as a society. So let's get to the end of the lecture. So these are some questions you might consider talking to your teachers and within each other about gene therapy. So could there be bad uses as well as good uses of gene therapy? And what are the prohibitive things that we think about? Well, clearly costs could be a big problem. And does this make it only available to the highly wealthy amongst us? Should people be allowed to have gene therapy to enhance their basic human traits? Height, intelligence, athletic ability? Could this be the new plastic surgery for the future? Could it also make gene therapy have another problem? Could the widespread use of gene therapy make society less accepting of people with maybe who are not perfect, So maybe the rich people are having gene therapy and the poor people can't afford it. Now that also raises another ethical question is would you want to do gene therapy on unborn children? Could you, for example, have germline gene therapy, and would that be acceptable knowing that you're now changing the course maybe of evolution of humans. So I recommend to you, your task is to see if you can find some examples of in gene therapy in use, and there are quite a lot out there, and there have been some bad ones as well, so you keep an eye out for those, and see if we can have a discussion about that next time we meet. So as a quick lecture summary, you should realise the human genome is very big. It's 3 billion base pairs of DNA. It's about 25,000 genes. We've learned how cells use the genetic code to make proteins. It's the central dogma of molecular biology. We've now used learned how we could use that information to create therapies, and how we could administer that to patients using vectors like viruses or plasmids. So hopefully we'll meet up next time, and we will speak soon.